Imagine a patient on an operating table right now. The surgeon, makes a critical incision, removes a suspected tumor, drops that piece of tissue into a plastic jar filled with some chemical preservative, and then hands it off to a nurse to be sent down the hall.
Right.
And we tend to think the most important part of the battle, like, the surgery just happened. But the truth is, what happens to that piece of tissue in the next 48 hours will completely dictate whether that patient lives or dies. So welcome to the command center of modern oncology.
Yeah, it really is a hidden front line because when we look at the entire landscape of cancer treatment today, the surgeon and the oncologist, they're basically executing a strategy that is entirely designed and dictated really by the pathologist's laboratory.
Exactly. And that brings us to our mission for today's deep dive. Because whether you are, say, a medical student prepping for boards, or a young professional entering the oncology space, or just a fiercely curious self-taught learner, we are unpacking a monumental paradigm shift today.
We really are.
We're pulling insights from this incredible textbook, Precision Cancer Medicine, Role of the Pathologist by Farhad Chassani, Ralph Huss, and Clive R. Taylor. And our goal here is to understand how pathology is basically undergoing its second major revolution in 200 years.
Yeah.
We are moving way beyond basic tissue diagnosis and stepping into this wild world of complex molecular biomarkers, artificial intelligence, and a whole slide imaging.
Right. And to really appreciate how massive this shift is, we have to look at the baseline first. I mean, for roughly 2 centuries, the absolute bedrock of pathology was just the light microscope and the H&E stain.
The chemotoxylin and eosin stain.
Exactly. Those are the classic pink and blue glass slides you see in literally every biology textbook. That stain, it actually invented the role of the surgical pathologist. You take a piece of tissue, you stain it, you look at the morphology, meaning, the shape and structure of the cells, and you make a diagnosis based purely on those visual patterns.
I've always kind of thought of the traditional H&E stain like a standard topographic map.
Oh, that's a good way to put it.
Yeah, it shows you the general landscape, right?
Yeah.
It gives you the tissue diagnosis, the grade of the tumor, how far to spread, which obviously gives us the stage. And that is incredibly crucial information.
Absolutely.
But, a topographic map only gets you so far. If you were trying to find a very specific person hiding in a specific forest on that map, topography just isn't enough. Precision medicine requires basically a molecular GPS.
And the necessity of that molecular GPS, it comes from a pretty dark history in cancer treatment, frankly. Because historically, oncology was fundamentally based on population-level statistics.
Right.
A doctor might say, well, clinical trials show this specific chemotherapy regimen works for, say, 30% of people with this broad category of lung cancer.
Yeah.
You were just treating the population average.
And crossing your fingers.
Exactly. Crossing your fingers that your patient just happened to fall into that lucky 30%.
Which is an utterly terrifying prospect when you're the patient sitting in that chair. I mean, you don't want to be a statistic. You want to know if the actual poison they're pumping in your veins is going to kill your specific tumor.
Precisely. And that's why the completion of the Human Genome Sequencing Project back in 2003 changed everything. We realized, we don't have to treat lung cancer as this generic monolith anymore.
Wow, yeah.
We can seek out specific genetic abnormalities or molecular lesions within an individual's unique tumor. We are treating a specific mutation that is driving that specific patient's disease.
But if we're going to use this molecular GPS, we have to actually locate the specific signals the tumor is giving off, right?
Right.
And the underlying mechanism here relies on identifying biomarkers. We are looking at the DNA, the RNA, or the protein level. And we do this using technologies like PCR.
Polymerase chain reaction.
Right, which essentially photocopies DNA over and over to find genetic typos.
Oh.
Or we use NGS, next generation sequencing, which to me is like speed reading the entire genetic instruction manual of the tumor.
It really is. Or, moving up from the genetic code to the actual physical structures, we look for abnormal proteins using IHC, which is immunohistochemistry.
Okay.
But here is where the terminology becomes clinical reality for you, the learner. In precision medicine, we have to categorize these biomarkers to know how to act on them. We have prognostic biomarkers, which tell us how aggressive a disease is likely to be, totally regardless of treatment.
Right, just a baseline forecast.
Exactly. But more importantly, we have predictive biomarkers, and these tell us if a tumor will respond to a specific drug.
I want to pause on that because the regulatory side of this gets complicated. A major focus in the source text is the difference between a companion diagnostic and a complementary diagnostic.
Yeah, that's a huge
Let
distinction.
me see if I have this right for the students listening. A companion diagnostic is kind of like a bouncer at a really exclusive club.
Mm-hmm.
If you don't have the right ID, like the positive test, you absolutely cannot get in to get the drug.
That's a perfect analogy.
Whereas a complementary diagnostic is more like a highly recommended Yelp review. Like, it's really good to know, it helps you make a choice, but nobody is legally stopping you from entering the restaurant.
Yeah, that's a very accurate way to frame it. A companion diagnostic is a strict FDA-mandated prerequisite. And the watershed moment for this was actually in 1998 with a drug called Herceptin.
Oh, right, for breast cancer.
Yes, it was designed to target the HER2 protein. And the FDA did something totally unprecedented. They approved the drug, but alongside it, they approved the Hercept test. The test and the drug were permanently legally linked.
Wow.
If a patient's tumor tests negative for HER2, giving them Herceptin is practically medical malpractice. It will not work, and you are only exposing them to severe cardiac toxicity for no reason.
Okay, so HER2 is a classic companion diagnostic. But it's not just breast cancer. We see this alphabet soup of markers across all of oncology now.
Oh, everywhere. Let's break some of those down so they aren't just, letters to the people listening. In breast cancer, beyond HER2, we look at ER, the estrogen receptor, and PR, the progesterone receptor.
Okay.
These are essentially docking stations. If a tumor has them, it means the cancer is using the patient's own hormones as fertilizer to grow.
So if we know that, we can use a drug to basically block the hormones and starve the tumor.
Exactly. Or, in anaplastic large cell lymphomas, we look for the ALK protein. When that's mutated, it acts like a light switch stuck in the on position, constantly telling the cell to divide.
And there's KRAS too, right?
Right. In colorectal cancer, we look for the KRAS mutation, which functions really similarly. It's a broken off switch in the cellular growth pathway.
But, identifying a broken off switch sounds straightforward in theory. Like, you run the test, you find the KRAS mutation, you prescribe the right drug.
Yeah.
Done.
If only it were that simple.
Right, because the deeper you get into the pathologist's reality, you realize that taking a piece of living human tissue, preserving it, and then extracting a reliable molecular answer, it's a complete minefield.
It really is.
If a patient's life depends on finding a single mutated protein, how do you even stop that protein from degrading between the surgeon's scalpel and the laboratory slide?
You have to control the environment flawlessly. And this brings us to the concept of the total test approach. We have to guarantee that the signal we see on a glass slide actually accurately represents the biology that was happening inside the patient's body hours ago.
Okay.
We break this down into 3 phases: pre-analytical, analytical, and post-analytical.
Let's start with the pre-analytical phase, which is essentially the garbage in, garbage out phase.
Yeah.
If you ruin the tissue here, literally no amount of advanced technology will save you.
None. And shockingly, this is where the vast majority of errors actually occur. The pre-analytical phase involves specimen acquisition, getting the tissue out, and then transport. And this introduces 2 terrifyingly critical windows: warm and cold ischemia times.
Break the mechanics of that down for me. What is actually happening to the tissue during those windows?
Well, warm ischemia is the time the tissue is deprived of its normal blood supply while it's still inside the patient's body during the surgery.
Oh, because the surgeon clamps the blood vessels, but the tumor hasn't been physically removed yet.
Exactly. Then, cold ischemia is the time between when the tissue is removed from the body and when it is finally placed into a chemical preservative in the lab. During both of these windows, the tissue is quite literally suffocating.
Wow.
Without oxygen, its proteins and its RNA begin rapidly degrading.
So it's like trying to bake a really delicate souffle, but your eggs are actively expiring on the counter while you run around looking for a whisk. If that cold ischemia time is too long, the molecular GPS signal you're looking for just vanishes before you even start the test.
It just disappears. It's gone. And once the tissue finally hits the preservative, which is usually a chemical called formalin, we enter the critical step of FFPE. That stands for formalin-fixed paraffin-embedded tissue processing.
Okay, and what does the formalin actually do?
Formalin's job is to create chemical cross-links between the proteins inside the cell. It essentially freezes the cellular structure in this tight molecular web so it doesn't rot.
But there is a massive catch here regarding the timing, right?
Huge catch. Total fixation time is this delicate tightrope. If you underfix the tissue, the web isn't strong enough, and the core of the tissue just rots anyway. But if you overfix it, those crosslinks become so dense and so tight that the testing antibodies we use later in the lab simply cannot penetrate the web to find the biomarkers.
It's like trying to read a letter that's been sealed inside a block of solid concrete.
which necessitates a process called antigen retrieval. We literally have to use heat and chemicals to carefully boil the tissue.
Wait, boil it.
Yeah, just enough to break some of those concrete crosslinks, letting the testing antibodies in, but without melting the delicate morphology of the cells themselves.
That sounds like an absolute impossible balancing act. But okay, let's assume the lab survives the pre-analytical phase. The tissue isn't garbage.
Yeah.
We move into the analytical phase.
Right.
And this is where a major conceptual shift is happening, according to the text. We're taking immunohistochemistry IHC, which involves painting proteins with antibodies linked to a dye, and we are forcing it to evolve.
We are?
It used to just be qualitative. Like, the pathologist looked at the slide and said, yes, it's brown, the protein is there. But now, we're forcing it to be a strict quantitative assay.
Yeah, the era of yes or no is completely over. The oncologist doesn't just need to know if the protein is there, they need to know exactly how much is there, down to the specific percentage.
Because the amount changes the treatment.
Exactly. The amount of protein expression directly dictates the dosage, or even the entire choice of therapy. And to achieve that, labs have to choose their methods incredibly carefully. They can build lab-developed tests, or LDTs.
Which are like custom homebrewed recipes, right?
Yes, custom recipes validated internally by that specific lab. Or they use ready-to-use, RTU tests, which are standardized FDA-approved commercial kits that are more plug-and-play.
And beyond just painting proteins with IHC, we're now probing the actual genetic blueprints inside the intact cell using ISH in situ hybridization.
Yes, and this is a beautiful piece of technology. While IHC looks at the finished product, the protein ISH looks directly at the DNA or RNA. The sources actually highlight a specific technique called RNA-scope.
I was reading about that. It's fascinating.
It is. We use custom molecular probes that bind to specific individual RNA molecules inside a single cell, and then they literally light up. It allows us to visually confirm if a specific gene is actively being transcribed into a biological command at that exact moment.
Okay, so the lab has flawlessly preserved the tissue. They've run this incredibly sensitive molecular assay. Now we hit the final hurdle, the post-analytical
The
phase.
human element.
Exactly. This is where scoring and interpretation come back in. And frankly, evaluating some of these new biomarkers, it sounds like we are asking the impossible of our pathologists.
Oh, we really are. It is pushing human visual interpretation to its absolute breaking point. Let's look at PD-L1, for example.
Right.
This is a crucial biomarker used to predict if a patient will respond to these revolutionary immunotherapies. Now, with older markers like HER2, you just look at the tumor cells and see if their borders are stained. Easy enough.
Right.
But evaluating PD-L1 requires the pathologist to score its expression on incredibly diverse cell types simultaneously. You have to look at the malignant tumor cells, and then you have to separately evaluate the surrounding immune cells like lymphocytes and macrophages in the exact same viewing field.
All right, let me make sure I'm visualizing this correctly for the listeners. We're asking a human being to look through a microscope eyepiece, visually distinguish between a tumor cell and a tiny immune cell, and then mentally keep a tally of the molecular expression across potentially millions of cells on a single glass slide.
Yes, that is exactly what we're asking.
That is absurd.
Okay.
I mean, human eyes get tired. We get distracted. Two different doctors looking at the same slide on a Friday afternoon are inevitably going to come up with slightly different percentages.
And you've just hit on the exact catalyst for the fourth industrial revolution in pathology.
Uh-huh.
Human visual interpretation is inherently subject to fatigue, subjectivity, and variability. And when a single percentage point difference in a PD-L1 score dictates whether a patient gets a life-saving immunotherapy or a highly toxic chemotherapy, human variability is just no longer acceptable.
Which ushers in digital and computational pathology.
Yeah.
We're finally moving away from the 200-year-old glass slide on a light microscope.
Exactly. The fourth industrial revolution is defined as the fusion of the physical, digital, and biological worlds. And in this field, it starts with whole flight imaging, or WSI.
Okay.
We take that physical glass slide and scan it using incredibly high-resolution scanners. Suddenly, the tissue is no longer trapped on a piece of glass. It's a massive, highly navigable digital file.
And this creates what the authors call a total integrative pathology problem, because it's not just a picture on a screen anymore. The pathologist is now looking at morphological data from the scan, genomic sequencing data from the NGS, proteomic data from the IHC. I mean, it is the ultimate big data problem.
It really is. The pathologist is no longer a simple visual inspector reporting on what a tissue sample looks like. They have to integrate millions of disparate data points to generate one cohesive, actionable clinical report for the oncologist.
I love the term the book uses to describe this new role. The pathologist is basically upgrading from a visual inspector to a knowledge engineer.
Yes, I love that too.
They're less like a detective with a magnifying glass and more like the conductor of a massive digital orchestra. But, to engineer that much data, a human needs a co-pilot. Enter artificial intelligence.
AI and machine learning are becoming totally non-negotiable tools. Think about that PDL1 problem we just discussed.
Right, the tallying.
AI algorithms don't get tired on a Friday afternoon. They can instantly and consistently count tens of thousands of cells across an entire digital slide to calculate a precise percentage. They can scan a sample and highlight tiny, microinvasive components of a tumor that a fatigued human eye would almost certainly miss.
But it's not just counting faster,
Yeah.
right? Because the text implies AI is actually seeing things we physically can't perceive.
Exactly. AI can evaluate the TME, the tumor microenvironment. It analyzes the intricate spatial relationships between the tumor cells, the blood vessels, and the surrounding immune cells.
Oh, wow.
It extracts geometric and spatial patterns that the human brain simply cannot process, and then it uses those invisible patterns to predict how aggressively a tumor will behave. Furthermore, AI is really the only way we can handle complex multiplex IF immunofluorescence stains.
What does that actually look like?
Imagine looking at a slide where instead of just one brown stain, you have 5 or 6 different fluorescent biomarkers glowing in different colors on the exact same tissue section simultaneously. A human can't disentangle that overlapping light. Our eyes just mix the colors. But an AI algorithm can separate and score every single channel flawlessly.
That is mind-bending. But, just when you think you have a handle on digital slides and AI, the deep dive into these sources offers a glimpse into emerging frontiers that sound like pure science fiction today.
Oh, it gets even crazier.
We are talking about abandoning the tissue entirely with liquid biopsies.
Yes, the liquid biopsy is a massive frontier right now. Instead of surgically cutting into a patient to get a tumor sample, we just do a simple blood draw.
Or just blood.
Yeah. We are looking for CTCs circulating tumor cells, which are whole cancer cells that have actually broken off the main tumor and are floating around in the bloodstream. We also look for ctDNA, cell-free tumor DNA.
And where does that come from?
As tumor cells die, they shed their mutated DNA into the blood, kind of like dead skin.
Which means you can dynamically monitor a tumor's changing mutations month by month without needing to perform repeated, highly invasive surgeries. You just draw blood.
It's incredible. It allows us to track the evolution of the cancer in real time. And on the imaging side, the physical limits of light microscopy are being completely left behind. We're moving toward technologies like MIBI multiplex ion beam imaging.
Okay, this was the part that absolutely stopped me in my tracks. Explain how MIBI works to everyone listening, because it doesn't even use light, right?
It doesn't. With MIBI, we tag the antibodies with specific heavy metal isotopes instead of traditional colored dyes. Then we place the tissue in a vacuum and literally vaporize it, layer by microscopic layer, using a focused ion beam.
Vaporize it.
Yes. And as the tissue vaporizes, a mass spectrometer reads the exact heavy metals being released, creating this impossibly detailed subcellular map of the proteins.
Vaporizing human tissue with an ion beam to map its proteins. I mean, come on.
Or consider Raman vibrational spectroscopy.
That's that one.
It uses lasers to measure the minute vibrational energy of chemical bonds within the tissue. Every molecule vibrates differently, creating a totally unique molecular fingerprint. It basically allows us to map the biochemistry of the tissue without using any stains or dyes whatsoever.
Okay, my brain is officially full. We have traveled from a 200-year-old pink and blue stain to literally vaporizing tissue with ion beams. So to synthesize all this instructional content and really cement it for the learners listening today, we are going to do a quick review exercise.
Application is the true test of knowledge, right? Let's see how these pieces connect.
All right, so to you, the listener, think about a patient presenting with metastatic colorectal cancer. In the context of precision medicine, why is it absolutely critical to test the tissue for a KRAS mutation before you administer an EGFR monoclonal antibody drug?
It's a great question.
And before we answer it, let's clarify a term from the sources that explains how we even know this. Prospective retrospective clinical trials. What does that jargon actually mean?
It's an important concept to grasp. A prospective retrospective trial means taking archived, preserved tissue samples from a clinical trial that happened years ago and applying a brand new molecular test to them today.
Oh, so you're looking back.
Right. You look back to see if this new biomarker could have accurately predicted which of those past patients survived and which didn't. And that is exactly how we discovered the critical relationship between KRAS and EGFR.
So based on that discovery, what is the mechanical answer to our review question? Why test for KRAS before giving an EGFR drug?
The answer lies in the biology of the cellular pathway. EGFR is a receptor on the outside of the cell that tells it to grow. The drug is designed to block that receptor.
Makes sense.
But KRAS is a signaling protein further down the chain, inside the cell. If the tumor has a mutated KRAS gene, it acts like a gas pedal that is permanently glued to the floor.
Ah,
It doesn't matter if the EGFR drug blocks the signal at the surface. The mutated KRAS protein is still screaming at the cell to divide. The mutation completely overrides the drug.
which means if you don't test for KRAS and you give that drug to a patient with the mutation, you are doing absolutely nothing to stop the cancer. You are only subjecting the patient to the toxic side effects of a completely useless treatment. And worse, you're wasting the most precious resource they have,
Yeah.
time.
Exactly. That is the ultimate realization of the companion diagnostic. If the molecular GPS tells you the bridge is out, you do not send the car down that road.
We have covered an immense amount of ground today, from the pre-analytical perils of suffocating tissue to the computational future where pathologists orchestrate massive data sets. But I want to leave you with a final, unaddressed concept to ponder on your own. It builds on everything we've explored, but takes it one step further.
I am always ready for a good thought experiment.
We open by establishing that the pathologist's laboratory is the command center of oncology. And for 2 centuries, the price of admission to that command center was a physical piece of solid tissue, the surgical biopsy. But consider the trajectory we are on. If liquid biopsies from a simple blood draw become so advanced that they can perfectly sequence all circulating tumor DNA, and if AI algorithms become sophisticated enough to predict treatment response entirely from that genomic code without ever needing to look at a physical cell,
Oh, I see where you're going.
will the traditional tissue biopsy eventually become completely obsolete? Will the anatomic pathologist who has spent 200 years looking at physical structures entirely transform into a data pathologist who only looks at code? Think about that the next time you picture the front lines of medicine. Until next time.